Hello students, the topic is fate map.
Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment.
A fate map is a diagram of an egg or blastula indicating the fate of each cell or region at a later stage of development. Fate maps are essential tool in most embryological experiments. They provide researchers with information on which portion of the embryo will normally become which larval or adult structure. The analysis of the fate of each blastomere after first and second cleavage is called cytogeny or cell lineage study. Construction of fate map. There are different methods. First one by observing living embryo. In some invertebrate, the embryos being transparent, having relatively few daughter cells that remain close to one another. It has been possible to look through the microscope and trace the descendants of a particular cell to the organ they subsequently form. This type of study was performed by Edwin G. Conkling 1905 in the tunicate Stylia partita, where the different cells contain different pigments. For example, the muscle forming cells always have a yellow color. Next is vital dye marking. Most embryos, however, do not have the facilities. That means they are transparent, few cells, different colors, etc. as described above in Stylia partita. It was in 1929 that work was able to trace the fate of different areas of amphibian eggs by applying vital dyes. These vital dyes stain the cells without killing them. Next is radioactive labeling and fluorescent dyes. A variation of the dye marking technique is to make one area of the embryo radioactive. A donor embryo is taken and grown in a solution containing radioactive thymidine. This thymidine base is subsequently incorporated into the DNA of the dividing embryo. A second embryo acting as a host embryo is grown under normal condition. The region of interest is cut off from the host embryo and is replaced by a radioactive graft from the donor embryo. The cells that are radioactive will be the descendant of the cells of the graft and are distinguished by autoradiography. Next is genetic marking. Radioactive and vital day marking have their own drawbacks such as dilution over many cell divisions and laborious preparation of slides. One permanent way of cell marking is to create mosaic embryos having different genetic constitutions. The best example of such a marking is to graft quail cells inside a chick embryo. By doing so, fine structure of maps of the chick brain and skeletal system can be made. This figure shows the fate map of Amphioxus. The fate map of Amphioxus can be traced at an early stage prior to the onset of cleavage. The future endodermal cells lie at the vegetal pole and would subsequently form the floor or hypoblast of the blastula. The area at the animal pole would form the presumptive ectodermal cells. The ventral grey crescent area at the future posterior end of the blastula in between the future ectoderm and endoderm forms the future mesoderm. Another area, the dorsal crescent in between the ectoderm and endoderm on the anterior side, gives rise to the notochord and neural cells. The presumptive ectodermal, mesodermal, notochordal and neural cells would subsequently form the epiblast of the blastula. This is the fate map of frog. The figure A is the lateral view, B dorsal view and C is the exterior view showing a thin outer layer of presumptive endoderm overlies the presumptive mesoderm in the marginal zone. The blastula of Xenopus at the 32 cell stage gives no indication as to how the different regions will develop. However, by following the fate of the individual cell or group of cells, the fate map of the blastula can be made. One way of making the fate map is by staining the various parts of the early embryo with a lipophilic dye. Another sophisticated way of labeling the blastomeres is by injection of high molecular weight molecules such as rhodamin labeled dextran, which cannot pass through cell membrane and are therefore restricted to the injected cell and its progeny. These cells can be easily detected later under a UV microscope. The fate map shows the presence of yolky macromeres at the vegetal pole, which give rise to endoderm. Depending upon the position of the blastopore, the endodermal area can be divided into subblastoporal and suprablastoporal endoderm. The cells toward the animal pole give rise to the ectoderm, which becomes further subdivided into epidermis and the future nervous tissue. The epidermal ectoderm forms at the ventral side of the animal hemisphere, while the neural ectoderm forms the dorsal side. The mesoderm forms a belt-like region known as marginal zone around the equator of the blastula. The mesoderm becomes subdivided along the dorsoventral axis of the blastula. The most dorsal mesoderm give rise to notochord. From this, ventrally the mesoderm is differentiated by the somites, which give rise to muscle tissue, lateral plate, which contain heart and kidney mesoderm, and blood islands. In Xenopus, a thin outer layer of presumptive endoderm overlays the presumptive mesoderm in the marginal zone. This shows the fate map of chick. In chick, the three germ layers of the embryo proper is formed by the epiblastic cells. The epiblast also forms a considerable amount of extra embryonic, that means mesoderm, membrane. The fate map of chick reveals that the cells of the epiblast are organized around the notochord and nervous system. The neural ectoderm is present as a knob-like structure facing towards the anterior side. The cells at the anterior part of the epiblast form the ectoderm, while the cells at the posterior side give rise to mesoderm, that means body proper, endoderm and extra embryonic mesoderm. Uses of fate map. The fate map of organisms is helpful in tracing the morphogenetic movements of the cells and the ultimate positions they take up. However, they tell us nothing about the tissue developmental potentiality during morphogenesis. Dear friends, thank you for watching. Don't forget to leave a like and please subscribe my channel.